Hello, this paper comes to you from Sandy Kinghorn and his father Brian, and we're talking about management of inbreeding and coancestry in breeding programs. And the first thing to note is that inbreeding is not heritable. Inbreeding is to do with your heterozygosity, and you cannot fit that into one sperm or egg, and therefore if you select for low inbreeding, well, these individuals can't transmit that low inbreeding to their progeny, so you only have a transient effect. On the other hand, coancestry is heritable. Coancestry here we're taking as the mean coancestry between an individual and the rest of the population. So if you take an animal which is largely new blood and use that animal for breeding, its progeny will inherit half of that new blood effect, and therefore coancestry is heritable. That has lasting effects to increase diversity and reduce inbreeding in the population in the long run. So inbreeding leads directly to loss of genetic diversity through this old formula. And we can reduce the rate of increase in inbreeding in three ways. One is to use more individuals as parents, and secondly is to select individuals that are on average less related to each other across the selected group. And lastly, we should allocate more matings to individuals that are less related to the rest of the population. And taken together, these three things constitute breeding for low coancestry in the parents that you select, so low parental coancestry. So in our paper we do population simulation using PopSim with a rather small population in order to have some notable effects. And here's an example of PopSim being bred over a number of years. Now of course PopSim has to make animal selections and mate allocations in each year of the simulation, and for that it launches and uses the program MateCell. I'm rather going to assume that you have some understanding of how MateCell works. Here is parental coancestry on the horizontal axis, and we want to be over to the left here with lower coancestry, higher diversity, and we want to be higher here with higher progeny index. So the policy for managing parental coancestry is managed by target degrees, in this case 10, 20, 25 degrees. And we're going to have treatments of 0 degrees, 25 degrees. Here you can see progeny inbreeding, and we've asked MateCell to manage inbreeding such that you don't have close relatives mated. In this case we've got full sibs, father-daughter and mother-son, and how they pan out in this exact data set, and MateCell is automatically avoiding inbreeding. So that's one of our treatments, is to avoid mating of close relatives. Otherwise we put a weighting on progeny inbreeding over here. So that is how we are invoking our treatments. Let's just look at what the treatments are. So for coancestry we have 0, 25, 45 and 75 degrees, and for inbreeding we're using weightings of 0, 1 and 100 against progeny mean F. And if you want to look at how these actually compete with each other, you need to know how we scale target degrees, and you can read about that in the manual at matecell.com. So let's move over to look at results, and the top two graphs relate to 0 target degrees, meaning that we're not putting any emphasis on reducing parental coancestry, and then 25 degrees for the bottom two. So in the top left graph you can see that putting a weighting of 0 on progeny inbreeding allows progeny inbreeding to increase over the years in the simulation, 20 years, and we managed to reduce progeny inbreeding increasingly by applying stronger weights and or, in the yellow line here, the policy to avoid close inbreeding. For 25 degrees we get the same sort of pattern but less difference, because we're now putting a fair bit of emphasis into genetic gains as well. And the two graphs on the right hand side are for progeny index or genetic gain. And firstly we can see that aggressive avoidance of inbreeding actually affects which animals are selected, and therefore genetic gain goes down. The other policies are sufficiently moderate that we do not get so much change in which animals are selected. However, where we do have higher inbreeding, then there does seem to be an effect to reduce gain, and this may be an effect of reduced genetic diversity because of more co-loss of alleles. And that effect's not evident when you're controlling coancestry here at 25 degrees. So there is an improvement here for policies that have got a moderate weighting against inbreeding. A key question is whether reducing inbreeding also reduces coancestry. So this graph has actually got 4 lines on it. The green ones are 2 treatments which are essentially superimposed. These 2 lines here, the red and the orange, are the inbreeding results for 2 different policies. And the orange policy is to have a weighting against inbreeding, and the blue policy is to not do that. So you can see that, of course, by having a negative weighting on inbreeding, it has an impact to reduce inbreeding throughout the course of the simulation. And of course, one thing to note is that the inbreeding levels tend to be lower and delayed compared to coancestry. And that is because the inbreeding in an individual is equal to the co-ancestry in its parents. So there's one generation of delay, and there's another effect that you can read about in the paper. However, the important thing here is that applying a moderately negative weight on inbreeding has no effect at all on the progression of co-ancestry in the population. There is a minor difference in there, but it's superimposed in this graph. The bottom line here is to look at genetic merit on the y-axis over the period of time of the simulation. So if we have a strong weight against inbreeding alone without controlling co-ancestry, largely because of selecting individuals that are not competitive in order to achieve low inbreeding, we're getting a much lower selection response. We get a much better selection response if we moderate that pressure against inbreeding in the progeny, still without controlling co-ancestry in any way. So when we add moderate control against co-ancestry with target degrees 25 and a weighting of 1 against inbreeding, we find that we get 12.2% improvement in genetic gain. Here it is here. But probably more importantly, we're getting more than 60% increase in genetic diversity. And that's one of the main reasons for managing co-ancestry, not just genetic gain, but the fact you have sustained genetic diversity for long-term gains. So we looked at this for target degrees 25 and a weighting against progeny inbreeding, just moderate effects. And we found that across all different combinations of treatments, managing co-ancestry was 4.9 times more important than managing inbreeding as far as improving genetic gain in the long term is concerned. And for improving genetic diversity in the long term, managing co-ancestry was 7.7 times more important than managing inbreeding. So co-ancestry management is the most important thing to look at. Manage that frontier on mate cell if you're using mate cell, or otherwise pay attention to your optimal contributions to manage co-ancestry well. Inbreeding is just a little extra bit. However, the best strategy is to pay attention to both co-ancestry and inbreeding. Thank you very much. Any questions?